The plasma membrane is made up of several organic molecules. I will concentrate on two of them in this presentation. The first molecule is a lipid. It is called a phospholipid because instead of the usual 3 fatty acid chains, there are only 2. The third bonding position is with a phosphate group. The phospholipid molecule is divided into a head region and a tail region. The head region is the glycerol and phosphate group. The tail region is the fatty acid chains. The head is hydrophilic, attracted to water. The tail is hydrophobic, repelled by water. It is this property which allows for the unique molecular alignment that makes the plasma membrane. If a droplet of phospholipid was put into water, a mycele would form. A mycele is a
Well, hi there! I'm a Golgi apparatus. I am found in most eukaryotic cells and most frequently near heavy protein concentrations. My function is to first receive molecules sent by the endoplasmic reticulum. I modify those molecules and package them into complex molecules. Some are stored, but most are launched and delivered to various destinations recruited out of the cell. But I bet you'd like me to clarify this, so I brought in a special guest, Mr. Post Office.
Hello, I'm the Post Office. I work a lot like my friend the Golgi apparatus. Just like the Golgi apparatus, when people come in with their mail, we receive it, we stamp it, and we ship it off to another place in the world.
Vesicles are sent from the endoplasmic reticulum to me, the Golgi apparatus, where we fuse. I then accept the proteins they empty. I modify them and transport them to their final destinations.
Just like how I fuse all the mail together, then ship them off to their final destination, and also modify them by stamping them as they pass by.
Lysosomes. What is a lysosome? Lysosomes are small organelles filled with over 40 enzymes. Lysosomes are created by the addition of hydrolytic enzymes in the rough ER and are modified in the Golgi apparatus. The picture below shows the Golgi apparatus producing lysosomes. What is a lysosome? Lysosomes are small organelles filled with over 40 enzymes. Lysosomes are created by the addition of hydrolytic enzymes in the rough ER and are modified in the Golgi apparatus. The picture below shows the Golgi apparatus producing lysosomes. Functions of the lysosome. Lysosomes break down lipids, carbs, and proteins into small molecules that can be used by the rest of the cell. Lysosomes are also involved in breaking down organelles that have outlived their usefulness. Lysosomes are responsible for getting rid of the junk that might accumulate and clutter up the cell. The picture to the right shows lysosomes destroying food particles. Where are lysosomes found? Lysosomes are found in the cell's cytoplasm. Several lysosomes float around in the cytoplasm until they find used organelles, bacteria, or other things that need to be removed from the cell. History of the lysosome. The lysosome was discovered in 1949 by Christian de Dubois, a Belgian researcher. He discovered them when he homogenized some animal cells into various components by running them through an ultracentrifuge. After a few days, the level of a few enzymes rose significantly, implying that they were segregated in the cell and had not attacked any part of the cell before they were broken down. Lysosome diseases. Several diseases are traced from the dysfunction of lysosomes, like Tay-Sachs disease and Pompe's disease. Tay-Sachs causes you to become blind, deaf, unable to swallow, have speech deficiencies, and much more. Pompe's disease causes weak muscle strength and failure of the heart. Many of these diseases occur in infants and children.
Most of a eukaryotic cell's ATP synthesis occurs in mitochondria. A mitochondrion is a sac within a sac. the inner increasing surface area for ATP synthesis. Between the inner and outer membranes is the intermembrane space, a reservoir for hydrogen ions used for synthesizing ATP from ADP. The inner chamber, known as the matrix, is a soup of enzymes that dismantle fuel molecules in the Krebs cycle. The knobs are where ATP is synthesized. Simply put, a mitochondrion is an energy transformer, where energy in fuel molecules such as pyruvic acid is transferred to ATP. Carbon dioxide, the carbon end product of cellular respiration, diffuses out, leaving the cell through the plasma membrane.
The Nucleus and Company by Joe Meyer and Anthony Constantino. Here is a quick preview of the topic covered today by a well-renowned scientist who wishes to remain anonymous as he is currently being hunted down by the Russian mafia for reasons unknown.
The nucleus is often referred to as the control center of the cell. Since this structure ultimately directs the cell's many activities, the nucleus contains the macromolecule vital to all life, deoxyribonucleic acid, or DNA. The nucleus is surrounded by a nuclear membrane, which is composed of a double layer of lipids and proteins. The nuclear membrane controls access to the instructions encoded within the DNA. The DNA found in the nucleus contains coded messages called genes that each cell uses to make thousands of different proteins, enabling it to live and to reproduce. The instructions encoded along the DNA are eventually passed on to successive generations of cells as each parent cell divides. When cells begin to divide, the chromatin, containing DNA and protein, forms rod-shaped bodies called chromosomes. Chromosomes are responsible for carrying the hereditary information to the next.
The nucleus has four major functions. They include storing genetic material, such as DNA. It also acts like the brain of the cell, controlling all of the functions of the individual organelles surrounding the nucleus. All expressed traits of an organism originate from the DNA that is protected in the nucleus. Here is a lovely diagram of the nucleus and its suborganelles. These organelles include the nuclear envelope, consisting of an outer and inner membrane, the nucleolus, located in the center of the nucleus, the nucleoplasm, which is a gel-like substance between the nucleolus and nuclear envelope, chromatin, which is suspended in the nucleoplasm, ribosomes, which pepper the outer membrane, and nuclear pores, which are holes which allow substances, such as mRNA, passage through the envelope. This short, action-packed clip illustrates the idealistic eukaryotic animal cell. This shows the relationship and scale of the nucleus to the other organelles in the cell. This is a close-up view of the nucleus, with its nuclear envelope and pores. The nucleolus is now highlighted in yellow to show its ideal location. Next, the nucleoplasm is highlighted, which consists of most of the nucleus and suspends the chromatin. Although chromatin is located in the nucleoplasm, the majority of this nucleic acid is found, densely packed, in the region known as the nucleolus. The nucleolus is made of densely packed proteins and nucleic acids. It is located in the center of the nucleus and is the site of transcription of DNA onto the mRNA. This mRNA leaves the nucleus to join with ribosomes and create proteins. These ribosomes are made inside the nucleolus. The nuclear envelope protects and encloses the nucleus. It is made of a double lipid bilayer. This envelope consists of an inner and outer membrane. The outer membrane has ribosomes scattered around its surface. There are also nuclear pores which penetrate the inner and outer membrane. This allows for the transport of materials, specifically mRNA, in and out of the nucleus. Please enjoy our fire.